A very good afternoon to everyone. On behalf of Director CSIR NIST, Dr. G.N. Sastry, I welcome you all for the Vertical 1 session. And due to unavoidable circumstance, today the Vertical 1 speaker, Dr. Indra Ghosh, couldn't deliver the lecture. So instead of her, now we have Dr. Kaushik Lakkaraju, who has vast industrial experience in molecular modeling with us. On behalf of DDH 2020 training team, as well as our leader, Dr. Sastry, I thank you, sir, for accepting our invitation to deliver this talk in a short notice. Dr. Kaushik is a senior research investigator in small drug discovery group at Bristol, Myers 2. He is a computational chemist with special interest in MD simulations, multi-scale modeling, and small molecule drug design as well as development. He gets involved in several in silico projects such as lead identification, hit to lead optimization. With this small introduction, I request Dr. Kaushik to deliver the lecture on site identification and beyond. Sir, please.
Okay, thank you everyone for coming, and thank you for the introduction and for the invitation. I just want to make sure you can see my slides, okay?
Sir, you have to share it again.
Okay. Okay, can you confirm that my slides are visible? Okay.
Yes,
great.
yes, fine, sir.
All right, so what I want to talk about today is this fragment-based simulation approach called SILKS, and what we can achieve in a drug design using a method like this. So it originally started off as a method that enables us to identify potential small molecule binding sites on a protein surface. But as we spent more time with the method, we realized there's a lot more data that can be harnessed here. So we'll be looking at all the potential applications in drug design that can be addressed using a method like SILKS, which uses fragments in its simulation strategy. So we could be looking at applications like lead optimization, applications like virtual screening, understanding protein-protein interfaces, and so on. So through the talk, the way this is laid out is we'll first look at SILKS as a method, the underlying details about how this method works. We'll be looking at a small example of how SILKS works in the context of identifying new chemical matter, and some of the more recent developments with respect to what's happening with the methodology itself. So when you look at typical structure-based approaches in the context of potency evaluation, a typical observation is that the accuracy of prediction for a given method and the speed of calculation oftentimes don't go hand in hand. So for instance, when you look at methods like docking, right? They're fairly quick at estimating a given binding pose, but the scores that are associated with docking oftentimes have very poor or modest correlations with binding affinity. On the other end of the spectrum, you have methods like FEP, which are FEP standing for free energy perturbation, which are thermodynamically very rigorous, right? But the amount of time it takes to evaluate this calculation makes it less feasible for evaluating a large library, right? So in terms of method development, oftentimes the motivation is to find a way in which you can combine the best of both the worlds. And another observation is oftentimes as you increase the rigor, it usually is accompanied by an increase in the conformational heterogeneity that is sampled by a method in its calculation. So so a strong motivation oftentimes is finding a way in which you can bump up the speed of a calculation, as well as improving accuracy. So what we're trying to do is to combine the best of both the worlds with respect to speed and accuracy for potency prediction, so as to find a way so that the method can meaningfully influence a drug design cycle, for instance. So in that context, what is SILCS? SILCS is an acronym for site identification by ligand competitive saturation. What it is this mixed solvent simulation strategy that's useful for affinity mapping of different fragment probes across an entire protein surface. So what really happens in SILCS is this. Say you have a structure of a protein of interest. It could be bound to a small molecule of interest, or it could be APO. But we take the APO form of the protein, put it in a solvent box consisting of a dilute aqueous mixture of different probe molecules or fragment molecules that oftentimes represent the different functional groups of interest in a ligand. So for instance, you could have benzenes representing aromatic functional groups in the ligand. You could have propanes representing aliphatic functional groups in the ligand. You could consider different types of hydrogen bond donors and acceptors in the form of formaldehyde, methanols, imidazoles, and so on. You could also have charged acceptors and donors in the context of using ions like methylammonium and acetates. So we take these representative fragments or small molecules and put them in the solvent box along with the protein. And then we run out this really long molecular dynamic simulation. So through the molecular dynamic simulation, what we then do is to map out the affinities of these different small molecules on the protein surface. These are called frag maps, and we'll be looking at in the next few slides what these frag maps are and how are they calculated. But essentially, that's what we're doing in SILCS. We map out the affinity of these fragments or these small molecules on the protein surface, and then try to use that information to understand the kind of a small molecule that would bind at a given site on the protein surface. Now, straight off the bat, the method has multiple advantages, as you can see. Given the fact that we're running this long time scale molecular dynamic simulation means that there's a inherent accounting for conformational ensemble associated with that protein instead of oftentimes a single crystallographic conformation. This simulation box consists of water and all of these different diverse probe molecules. So through the MD simulation, what we are accounting for is the inherent competition between these different fragments and water to a given protein size. So what we're looking at is inherently who is going to win that competition for a given site. Registers the best affinity for that given probe. And like I said, given the fact that there's explicit water within the simulation, accounts also for the desolvation that accompanies a ligand binding on a protein binding site. So what are these frag maps and how do we get these affinity patterns? So consider the situation on the top left first. Say you have just empty solvent box, right? Just water. And in the solvent box, let's consider a given probe molecule, like for instance, methanol. And let's say you take this solvent box and you run out a really long MD simulation. Now, through the MD simulation, what you then do, or at the end of the MD simulation, what you can then do is if you bin out the solvent box into voxels or grids, and then count the number of times a methanol molecule has visited that grid or a voxel, you will see that there's no difference in the count no matter where you take that counting. Now that's primarily the effect of equilibrium, right? Like you take a given concentration of methanol and you run your simulation long enough and you count the number of times a methanol has visited a given site in the solvent box. That count would be exactly the same no matter where you make that measurement. Now bring in the protein of interest. right? So in the same solvent box with the same methanol and water, now you have a protein of interest, and you rerun the MD simulation. At this point, when you recount, the count of the methanol in each of these grids is now going to differ based on how much of an interaction exists between the protein surface and that given methanol. There could be certain sites where the protein surface doesn't like the methanol. There could be certain sites where the protein likes the methanol. Or in other words, now when you redo this exercise of binning down the entire simulation box into these grids or voxels, and within those grids and voxels, if you count the number of times a methanol has visited, that count is going to change depending on where you make the measurement. Closer to the protein surface, at least certain sites that like to make that hydrogen bonding, a methanol is likely to be registered for a longer period of that simulation time compared to the further you travel away from the protein surface. Those voxels are going to represent what looks like a the bulk phase part of the simulation, or when there's no protein here. Now, given the fact that you now have these counts of how the methanol is in the presence of the protein versus in the absence of the protein, you can take that ratio, right? So now that would give you the probability. And once you have that ratio, taking a ln and adding this prefactor of KT now puts you into the free energy realm. So what is grid free energy is essentially this. You are evaluating the free energy, or you're basically evaluating the probability of finding a given probe molecule in a given grid or a voxel or a subspace. So that is what is grid free energy. So you run this simulation twice. One is in the absence of the protein, one is in the presence of the protein. Excuse me. And then you normalize this. So you normalize the count that you've registered in the presence of the protein by the count that you would get in the absence of the protein, and that would then lead you to what's called as a grid free energy. So this is how SILKS is basically taking in and counting for or evaluating the affinity patterns of these different probe molecules on a given protein surface. Now, in the real world, right? So if this was a study that was done several years ago, I'm pretty sure this number has slightly changed, potentially even more biased towards this observation, that if you take a list of all therapeutically relevant proteins and bin them into different classes of receptors, there is this observation that's found that nearly 40% of all the drug targets try to target two classes of receptors, G protein coupled receptors and nuclear receptors. And both of these come with their own set of challenges. And a common theme across both of them is the fact that the small molecule binding site is often buried. I mean, in the case of nuclear receptors, it's completely blocked, at least in the crystal structures that we have on hand. And GPCR also, this is sufficiently buried. Now, how is this a challenge for a method like the free energy approach, or like any method that uses molecular dynamics, including SILKS, is this? So in the context of SILKS, in the context of evaluating these affinity patterns of the fragment, here's a challenge. The more buried the binding site is, the longer it takes for a fragment or a probe molecule to travel from the bulk phase of the simulation into the binding site of interest and back. And so what is happening is now we are rate limited by diffusion. If you rely on conventional molecular dynamics as a solution for evaluating how many times a given probe molecule visits a given protein site. And given the fact that we're now turning this into a free energy, means that this cannot be a single event. It has to happen over and over again before you have statistically relevant information to evaluate free energy in the context of number of times a fragment has visited a given site and then taken off. So that is the fundamental limitation of using a method like molecular dynamics. dynamics, in the sense that you're now rate limited by diffusion to evaluate affinity patterns for these fragments. So the amount of simulation time needed, if you use just molecular dynamics to evaluate these classes of receptors, to evaluate the affinity patterns of these probes to these classes of receptors, it climbs up exponentially. So what is our solution for this problem? Well, we use this method called as grand canonical Monte Carlo and couple that with molecular dynamics. Now, all of us are aware of grand canonical Monte Carlo as an approach. It's basically GCMC. There are essentially 4 types of moves that can happen in a GCMC: an insertion move, a deletion move, a translation, and a rotation move. And each of these moves are governed by a Metropolis criteria, which is fundamentally driven by the delta E and this factor B, which I'll get to in just a minute. But the Metropolis criteria is essentially driven by this delta E. And what is delta E? Is a change in interaction energy that a fragment or a probe molecule would feel as it moves between 2 different medium. So here's an instance, right? So this is a schematic. In gray is your protein site of interest, and each of these are your fictitious gas phase reservoirs. So now you can use the Metropolis criteria to evaluate what it takes for a probe molecule to move from this gas phase reservoir into the site of interest and back. So that those 2 moves can be classified into insertions and deletions, right? So an insertion is qualified as a move where you move a fragment or a probe molecule from a gas phase reservoir into the site of interest. A deletion is the same thing, except that now you're moving the fragment from the site of interest back into the gas phase reservoir. So if a probe molecule of interest doesn't like a given site on the protein, then the delta E would turn unfavorable, and it would prefer to go back into the gas phase situation. And the opposite is true. If a given probe molecule likes a protein surface, then it would move instantaneously from the gas phase reservoir back into the site of interest, right? So this is how we're using a Metropolis criteria to instantaneously move a probe molecule or a fragment molecule from this gas phase reservoir to the site of interest. This is potentially how GCMC would work. Now, in the context of a condensed phase simulation where there's explicit water, that throws in an additional challenge. And what is this challenge? Is that depending on the nature of your probe molecule, be they could be apolar, for instance. Like let's take a benzene molecule, or let's consider a methanol molecule. So depending on the nature of this probe molecule, in a condensed phase environment, 2 things can happen. One is there could be a shell of water around the probe molecule of interest, or the probe molecule of interest could be forming a network with the neighboring waters, again, depending on the nature of it. In each of these instances, the probe molecule likes that microenvironment in which it exists. And therefore, delta E basically skews you towards a point where an insertion move is almost never going to happen. And similarly, a deletion move is almost never going to happen if you base this Metropolis criteria purely on delta E, which is when this factor of B kicks in, where B is basically beta mu Ex plus ln M. What is mu Ex? The excess chemical potential. So in a ideal situation, the excess chemical potential is going to be the solvation free energy. The amount of effort it takes to move a probe molecule from the gas phase reservoir into a solvent phase. But in the context of evaluating or driving enough number of fragment moves from the bulk back into the binding pocket, we've observed that we now have to bump up or bump down this excess chemical potential in response to the number of fragments that you're observing. Here's an algorithm details of what's really happening. This is the paper that delineates all the details of the method, but what it does is briefly this. So we run some number of GCMC steps for all these different fragment molecules within that site of interest. And then we couple that GCMC with molecular dynamics. Now, it's important to continue to incorporate molecular dynamics in the context of evaluating the conformational ensemble or the conformational heterogeneity of both the protein and the fragment molecules in response to each other. So we do GCMC primarily to overcome the limitation of diffusion in molecular dynamics. But we continue to retain molecular dynamics to understand conformational heterogeneity, like I said, in the context of the probe molecule in response to the protein and backwards. So you run GCMC, you run molecular dynamics, and then what you do is to evaluate the number of fragment molecules that exist in that site in comparison to the number that should be existing in that site. So for instance, if the goal of this simulation is to observe what happens with 1 molar of a fragment molecule on a given protein surface, what you're doing is you're counting the number of fragment molecules that have eventually made it into that site of interest using a GCMC styled approach. And then you see if that count is lesser than or greater than the 1 molar of the fragment. Again, 1 molar is anecdotal. In reality, in GCMC, it's a much dilute mixture of 0.25 molar. But for the sake of understanding this metric, let's stick to 1 molar as an example. So now you count the number of fragment molecules that exist in this simulation box. If the number is less than 1 molar, you know that for some reason the fragments are not liking this solvent environment and they'd rather go back into the gas phase. So you kick in an artificial amount of work done in the form of delta mu excess chemical potential, or delta excess chemical potential is delta mu, and add it to the excess chemical potential and use that to drive your metropolis criteria. So you're artificially stepping on accelerator, for instance, in order to move enough number of fragments from their gas phase environment towards the condensed phase situation. The converse can also happen. There can be instances when a fragment molecule enjoys coming in and binding to the protein surface to the level that you could be having much more in excess of your target concentration. In which case, now you do the opposite. You apply a brake even more harder and you bump up the negative delta mu, so that you are now artificially yanking out or pulling out enough number of fragment molecules from the site, so that the target of evaluating how 1 molar of a given fragment or a given probe molecule samples the protein surface stays throughout the simulation. I mean, there are of course some more details of the simulation with respect to building grids, detecting the cavities, inserting the fragments into those cavities, and so on. But in the interest of time, let's move on. So I just want to update this fact that all of these is happening in a random fashion, in the sense that you bin out your entire simulation box into grids again. And within those grids, you make a note of where all cavities exist, and then randomly pick any of those sites or any of those cavity sites, and randomly pick any of these fragment types and bring those or try to bring a fragment from their gas phase into that, the gas phase cavity site, and evaluate using the metropolis criteria, where you're now bringing in this bias with respect to the work done in the form of this excess chemical potential, in order to accept a move, be it into the site of interest or away from the site of interest, and rinse and repeat. So you go through this process of a large number of GCMC steps, where you're now instantaneously sampling the ability of different probe molecules to come to the site of interest and back. And then through the simulation, you're now counting again the grid free energy, in the sense you're now taking an account of the number of times a fragment molecule visits a site of interest and normalize that with what would happen in a bulk phase situation. And then you go through with this process by accounting for molecular dynamics. So here's a real life example of how these frag maps would look like. This is obtained, this is again anecdotal, this is obtained from running a silk simulation with the protein tyrosine phosphatase 1B protein with this PDB2QBP. And you see how spatially different fragment molecules like to sit at different locations on the protein site. So here's a binding pocket of interest. Now, in the case of PTP1B, we know that this is a binding site based on crystal structures available of PTP1B bound to small molecules. Now, if you zoom into that site of interest, this is pretty much how spatially the different probe molecules have decorated this binding pocket. Again, obtained from the grid-free energy styled approach of evaluating these affinity patterns. The color coding is here. So basically green is for how the apolar fragments have decorated this pocket. Orange is how the charged, negatively charged acceptors have decorated this pocket. Cyan is basically how positively charged fragments of probe molecules have decorated this pocket. The fact that none of it exists basically means that there's nothing here. There is a small blob here. And then, of course, you have hydrogen bound donors and acceptors in blue and red, respectively, as to how they decorate this site. Now, when you bring back a ligand, and let's call this a reference ligand or a parent ligand, you actually see how beautifully the different functional groups in your ligand overlap with their respective frag map types that have been obtained from the independent silk simulation. So for instance, you have good overlaps between the acceptors in your ligand and the corresponding fragment types that have sampled this protein surface independently from the silk simulation. Similarly with the the apolar functional groups and the overlap between the apolar functional groups and the apolar frag map simulations that you've got. So straight off, by looking at this, you get a qualitative picture of using these frag maps. You get a qualitative picture of how these probe molecules are decorating a binding pocket, and from that you can have a sense of the kind of ligand that would be sitting in that site. And you can then think of optimization strategies where, for instance, now you can start growing out of this phenyl ring into reaching out, you can grow out another functional group from here to now reach into this site that has been marked by the negatively charged frag maps, right? So this is basically saying that this binding pocket tolerates having a negatively charged functional group because there is potentially a salt bridge to be useful, to be used in order to bump up the potency of this ligand, and so on. So for instance, you can start replacing these phenyl rings with potentially heterocycles that consist of donors and acceptors to map with these overlaps with these different donor and acceptor function groups. Now, this is still a qualitative picture, as in like you are loading your protein of interest, you're going into the binding pocket of interest, and then you are seeing how the functional groups or these probe molecules are decorating that pocket, and you're using that information to decide, okay, now how do I optimize this ligand? Can you turn those into quantifications? So we do have this metric called as the ligand grid-free energy. And what it is it's simply taking off from the grid-free energies that were calculated. So just to refresh your memory, grid-free energy is basically KT log of the probability of finding a probe molecule at a given grid or a given subspace in the simulation box, right? So now when you bring back your ligand of interest into that site, for instance, you can quantify or evaluate how good of an overlap exists between the different functional groups in your ligand and the corresponding frag map types that have been calculated or pre-computed from a silk simulation. The better this overlap is, the higher the reward with respect to giving a higher or a more favorable ligand grid-free energy score. And conversely, the poorer this overlap is, the less likely that ligand with this combination of functional groups likely to stick into that binding pocket. Of course, you don't have to restrict yourself to a single conformation. I mean, there could be instances when you don't have crystallographic binding pose information of how a ligand sticks to that binding pocket, in which case what we do is we run a second round of Monte Carlo sampling, except that this is different from the GCMC driven sampling of the fragments in the sense that, or the probe molecules, in the sense that now you take the full ligand and you have, again, you have a pre-computed map of how the different atoms in the ligand can be mapped back to the different pre-computed affinity patterns, right? So what we then do is we take these pre-computed frag maps and we run a Monte Carlo sampling of the ligand in the field of these frag maps, and then get an ensemble of conformations that the ligand could be taking in the binding pocket. And from that, we can then derive the ligand grid free energy. Again, this is a metric that was benchmarked almost 4 years ago, so pretty sure this number is even more better now. But this is a ballpark. This is giving you an idea that you can evaluate almost 1,000 ligands within half an hour in a single CPU core once you have pre-computed these fragments. So with respect to the Silk's approach of using fragments to run the simulation, that is the most expensive part of the calculation. You pre-compute those fragments, you run out the long time scale grand canonical Monte Carlo molecular dynamics simulation, get the frag maps, and once you have the frag maps, running this ligand grid free energy using those frag maps is almost instantaneous. So this is where that number is coming from. And for those of us who are familiar with more thermodynamically rigorous approaches, like free energy perturbation, for instance, we know that those approaches tend to come with their set of limitations or constraints, in the sense, like, for instance, if you're using the free energy perturbation style evaluation to see how best a ligand can sit into a binding pocket, that has to happen in the relative free energy realm. So you're considering transmutating a pair of ligands for that kind of a calculation. And even within those pair of ligands, that usually is accompanied by the two ligands can't be too dissimilar from each other. On the other hand, in the Silk styled approach, where you've pre-computed the fragment free energies, you now no longer have those kinds of constraints or those kinds of restrictions. In fact, you can bring in as dissimilar ligands as you want, and then just evaluate how good of an overlap exists between the functional groups in your ligand and the pre-calculated affinity patterns, which look at how the different, frag maps or different affinity patterns have decorated this pocket. If there's a good overlap, you give a good LGFE score. If there's a poor overlap, you just assign a poor LGFE score, and you use this for rank ordering. So this is an approach that can be used for efficiently driving an optimization study. So also the fact that each atom in the ligand maps back to the corresponding affinity pattern that has been independently calculated. We can also assign atomistic grid free energies, in the sense that you can determine individually what each functional group in the ligand is doing towards driving its full binding to the binding pocket. So these are such some of those atomistic grid free energy scores that you can assign to a given ligand. So this was also the time when we were considering running a rapid virtual screening study. So towards that, what we did was, along with using ligand grid free energy, we also explored this alternate pharmacophoric approach. So what are we doing in the pharmacophoric study is this. So again, here's a cartoon. Assume that the blue dotted line is your protein of interest. And of course, now you binned out the entire simulation box into voxels or subspaces. And you've calculated, you've pre-computed the grid-free energies from a Silk simulation. What we can then do is run a scan across across each of these voxels. And from the scan, you can then build a cluster of of where a given affinity pattern matches. So from those clusters, you can then come back and say, oh, this is a good pharmacophoric feature that's likely to stick into this site. So here's an example, right? Like, so this is a protein surface of interest. And this is how spatially the apolar probe molecules and the donors and the acceptors decorate this protein binding pocket. By running this clustering styled approach, we are then turning these frag maps into pharmacophoric features. So you turn this apolar blob, if you want to call it that, into a single apolar pharmacophoric feature. The neighboring donors then become a donor pharmacophoric feature, and so on and so forth. So of course, you can then, what you do is you would get a bunch of pharmacophoric features of the kind of ligand that is likely to sit there. And then you can pick, 6 pharmacophoric features, 5, 4, or whatever that is most relevant for that binding pocket. And if you have your vendor databases of the kind of ligands that you want to sample there, turn them into pharmacophoric features. You can then run a quick pharmacophoric screening of those ligands into that site of interest. So here's a benchmarking result where we took the HIV protease, ran silk simulation, turned those into pharmacophoric features, and took the DUD data set, which consists of both actives and decoys. And we were seeing how quickly each of these different methods like DOC, AutoDoc, and the pharmacophoric approach from the silk simulation, how quickly can they correctly identify actives from the decoys in that given set for this given binding pocket. And as you can see, the more the number of pharmacophoric features that you consider from this approach, of course, the better the accuracy. And you can see that quickly, the pharmacophoric approach seems to be doing a good job in picking out actives or separating actives from the decoys within a given data set for a given binding pocket compared to DOC and AutoDoc and so on. So this was very promising. The fact that you could get these pre-computed frag maps and then turn those frag map features into pharmacophoric features and then use the pharmacophoric features to drive virtual screening. So here's a quick example of where this worked, right? Like so now we've worked on those individual pieces of running out a silk simulation and then using that information. Here's an example of where that silk simulation was useful in identifying new chemical matter. So we applied these different moving parts towards understanding the binding pocket requirements of this GPCR, the beta-2 adrenergic GPCR, and then use that to see if we can come up with new agonists that would likely to bind here. So in the case of the beta-2 adrenergic GPCR, the advantage was that we had crystal structures available, and those of us who work in the GPCR space know that this is oftentimes challenging. But in the case of the beta-2, we were lucky to have crystal structures available. So in fact, there was one crystal structure that represents the active conformation, colored in orange, and there was another crystal structure representing the inactive conformation, colored in green. So what we did was we ran 2 independent silk simulations with each of these starting conformations. And I'll tell you why is this relevant in the next slide. But I do want to reiterate that reaching into the GPCR pocket is challenging by itself, but enabled by this approach, the grand canonical Monte Carlo coupled with molecular dynamics, we could quickly drive an efficient sampling of these different probe molecules into reaching that site of interest and out fairly quickly. Well, we still had to do 50 times 10, so a cumulative 500 nanoseconds molecular dynamics to account for the conformational heterogeneity. But you couple that with 100 million steps of GCMC, and now you have an efficient sampling that's thermodynamically relevant for each of these probe molecules into how they coat or decorate the binding pocket. So of course, this is where your protein is. The protein is surrounded on both sides by the lipid bilayer, and you have an extracellular binding or an extracellular solvent phase and an intracellular solvent phase, again coupled with these different fragments. So we run these simulations, and to our surprise, what we noticed was the binding pocket requirements for an active conformation protein is different from the binding pocket requirements for an inactive conformation of the protein, which kind of sort of makes sense in retrospect about the change in the thermodynamic peaks and the valleys as you move across these two conformations. But it was interesting that the silk simulation was able to detect the difference in the way a ligand would stabilize an active conformation versus an inactive conformation. So we thought, okay, hey, we now have these frag maps that have these differences in the way they coat an active binding pocket versus the way these frag maps map out the affinity patterns in an inactive conformation. So we thought we could use these differences to drive a screening study. So that's what we did. We turned these frag maps into pharmacophoric features and ran a pharmacophoric screen with some of the vendor databases, identified a shortlist that seemed to be sitting well into these sites. And then now from this point on, started running a screen that preferentially drives binding of ligands into the active or stabilizes the active binding pocket compared to the inactive binding pocket. And from that, of course, we come up with a list. We, of course, ran chemical clustering primarily towards coming up with heterogeneous chemical matter. In the top here are some of the previously known agonists that bind to the beta-2-adrenergic GPCR. And below here are the kind of compounds that come out from this silk screening approach. So again, like a quick glance shows us the diversity in the chemical matter that's coming out from this approach. Which is interesting by itself. But do these compounds work? Yes, emphatically yes, in the sense that what we did is we ran a bunch of ex vivo studies in the sense that we took these compounds, took out trachea from the mice and observed, you pre-constrict this trachea and observe how much of this tracheal relaxation is driven by the beta-2-adrenergic GPCR engagement by the ligands. And we see that 7 of the 15 compounds that were selected from the study do induce a tracheal relaxation via the beta-2-adrenergic GPCR mechanism. So this was a kind of a nice rounded story towards understanding how these different moving parts in silks came together towards identifying new chemical matter. So we had to use the grand canonical Monte Carlo, couple that with molecular dynamics to get good affinity patterns of the binding pocket, turn those affinity patterns into pharmacophoric features to drive an efficient pharmacophoric screening, and then use the ligand grid free energy as a further rank ordering process towards picking ligands that tend to occupy that binding pocket better. And then, of course, this was validated using this ex vivo studies. So one advantage of running the silk simulation is the fact that you have these frag maps that are pre-computed, and you can keep going back to these pre-computed frag maps to get new data or to get new information. And here's an example of that. So this is a silk simulation that we ran with the androgen receptor, and from the silk simulation, of course, you get the affinity patterns of these different probe molecules across the entire protein surface, including the fully buried orthogonal binding site or orthosteric binding site. Sorry, and just by looking at the orthosteric binding site, you know that the fragment patterns make a lot of sense. Like for instance, the apolar functional groups, actually the aromatic functional groups and the aliphatic functional groups, correctly overlap with the way a dihydrotestosterone binds into that site. So this by itself is an interesting find in the sense that the frag maps using the grand canonical Monte Carlo approach reached into that site, although completely buried, and sampled the binding pocket correctly. But this is also coupled with sampling the anti-protein surface. So now you can go back and see, hey, where else do we find interesting binding sites, and are those hotspots or potential binding sites? Can we reach into those using allosteric or treat them as allosteric binding sites and reach into those as well, right? So in that context, I don't know if this is quite visible, but marked by these black arrows further away from this orthosteric binding site are known allosteric binding pockets where flufenamic acid binds into those sites. So what we noticed is, if you go back, you actually do see a good overlap between the flufenamic acid's functional groups and how the affinity patterns were obtained for the different functional groups from the silk simulation at that site. And now if you use ligand grid free energy as a way to rank order these given sites, you actually see that the trend holds up. And what I mean is this. So again, like these frag maps are pre-computed already for the androgen receptor. So what you can do is to split the frag map for the anti-protein surface into subspaces, bring in a ligand of interest, in this case, a flufenamic acid and your dihydrotestosterone, and see how each of those ligands rank order with respect to the LGFE in each of those subspaces, right? So that's what becomes your x-axis with respect to a site rank. And see if the true binding site for that ligand is correctly rewarded by a good LGFE scoring. And that's what we notice here, that for instance, the flufenamic acid binding site here, which is the true binding site, does turn out to have the best LGFE score. So the trend is basically correct in the sense that the true binding pocket does register well with the best binding score coming out from the ligand grid free energy approach for a given ligand. So you can then use this as a way to identify potential binding hotspots, not only for an orthosteric binding site, but also for an allosteric binding site. Another very interesting development with the silk frag maps is to use this as a way to understand protein-protein docking. In the sense that, so if you think about this, right? So there's a vast number of drug designing applications, including novel modalities like using PROTACs, for instance, where you want to recruit a protein towards proteasomal machinery and so on, or towards understanding antibody binding to a given receptor. It's all driven by protein-protein docking, or the way the two proteins interface with each other. And if you consider the conventionally used protein-protein docking approaches, they almost always use crystallographic conformations. And we know that more often than not, when two proteins bind and talk to each other, there is a cooperative effect. There is something happening conformationally as the two proteins interface with each other that may not be represented in the individual crystallographic crystallographic structures that are obtained separately for each of those two binding partners. And given the fact that in silks, you're already accounting for conformational heterogeneity, oftentimes in the order of microseconds, when you're evaluating those affinity patterns, it's possible that this conformational change is already registered when you're getting those affinity patterns. So this was something that was tested out in this approach. So you take your two proteins of interest, run out separate silk simulations with each of these proteins, and get the affinity patterns. And once you get those affinity patterns, you can then see how those two patterns could potentially dock with each other by registering complementing ways in which the two frag maps bind. So for instance, if you have a patch of negatively charged frag maps on protein A and a patch of positively charged frag maps on a protein B, and then you can then consider permutations and combinations in which these two complementary frag maps are talking to each other and thereby engaging how the two proteins could potentially dock with each other. So that's what we do. We take the two proteins, we take the two protein maps, get the combined way in which the two maps talk to each other using a fast Fourier transform styled evaluation of the different poses that the two frag maps can take with each other. And from that, recalculate how the two protein binding partners are talking with each other. So this has, like I said, far-reaching implications. One such very interesting development or very interesting application where this is actually a work that's being, there's a paper that's being written just now, but what's happening is this. So, oftentimes when you design monoclonal antibodies or antibodies in general, a significant challenge is to preserve the stability of this antibody as it waits before we administer this to a patient. Right, so you need to find ways in which you want to preserve the stability of this protein as it sits on the shelf. So what people oftentimes do is to maintain this antibody in a solution that consists of excipients, which prevent this antibodies from, say, aggregating with each other and thereby becoming destable. But how do you find the right combination of these excipients for a given antibody, right? Like oftentimes that's trial and error. But what we noticed is in the case of silks, you can actually come up with a rational way of identifying those excipients that can be used for suspending this, this given antibody so that it stays stable. So how do you do that? You take the antibody of interest, you run out a silk simulation, and then from the silk simulation, you can then run a protein-protein docking to see potential sites where the two antibody partners could be binding with each other and thereby aggregating. So now you have those potential sites. And then now you take your list of excipients. In this context, I believe they went after all the FDA-approved excipients. So you take these excipients and you run that hotspot analysis to see where these excipients bind. And the places where the excipient binding matches or overlaps with where the antibody could be interfacing with its own binding partner, you can use that to rank order which excipients are likely to disrupt this binding process and thereby enabling stabilization, which is primarily how they've come up with designing excipients to help with stability of the antibody. Another very interesting study that's currently going on is this permeability approach, where if you have a given bilayer of interest, you can use the silk simulations as a strategy to understand permeability of which ligands are likely to pass through a given bilayer composition. So there's a lot of interesting ways in which you can keep going back to your affinity patterns to tap in and retrieve this new information for these different aspects of drug design. Right. So in the interest of time, I'm going to quickly summarize what we've seen so far, and then we can, of course, take lots of questions based on your interest in the given aspect, in the different aspects of the silks. So what is silks? It's basically this mixed solvent simulation strategy that's useful for efficient mapping of affinity patterns of these different fragment or probe molecules across an entire protein surface. GCMC-MD is this hybrid stimulation strategy that enables accurate sampling of all the protein surfaces, including the buried sites. Ligand grid free energy is the scoring technique that taps into frag maps that obtained from the silks to enable rapid rank ordering of ligands per binding site. Silks farm is this approach where you convert the fragments, the frag maps, or convert the affinity patterns into pharmacophoric features that can then enable rapid virtual screening. And then, of course, the pre-computed frag maps, we can always keep going back for getting new information. And what we notice is in the context of enabling identification of alternate binding sites. And of course, frag maps can also be used to guide the study of protein-protein interactions. And we saw an example where the silk's GCMC-MD was useful for characterizing differences in binding pocket requirements between the active and the inactive conformations of the beta-2 adenosyl GPCR. And we use those differences in the frag maps to identify new agonists targeting the beta-2-adrenergic GPCR. Here are a couple of patents that came out from this study. So I don't want to conclude by acknowledging all the different people that were involved in the data that you saw today. So the SILKS approach itself came out from Professor Alex Mackerel's lab at University of Maryland School of Pharmacy. And Prabhu, Keno, Wenbo, and myself, we were all involved in different stages of the SILKS development. Prabhu is now at Dassault Systems in BioVia. Keno is now a faculty at University of Bristol. Wenbo is a faculty now at University of Maryland School of Pharmacy. And what we noticed is that there is a lot of industry interest in trying to use these frag maps in their own individual drug design. So there's been a company called SILKS Bio, which took off from using the SILKS as an approach. So it's now turned into a product. And I worked previously with SILKS Bio with Sunwanjo there. And what we did was basically take this method and turn it into a product that can then enable rational drug design by different pharma companies. So I do want to thank people at Pfizer, my manager, Denny at Pfizer, with whom I interacted fairly extensively, and all these different computational chemists who gave critical feedback at a time towards finding the best ways you can turn the SILKS method into a product that can enable computational chemists to use this in the context of drug design. And then, of course, the beta-2-adrenergic GPCR studies, the compounds that were identified using the SILKS approach were tested by Professor Deepak Deshpande. He was previously with the University of Maryland School of Medicine. He's now at Thomas Jefferson University in Philadelphia. And then, of course, my current employer, Bristol, my Squibb, and my managers here, Karen Rossi, Steve Johnson, and Dan Cheney, at various points where they were involved in evaluating these fragment-based simulation approaches. So with that, I thank you all for your time, and of course we can take questions now.
Thank you, sir. Sir, am I audible?
Yes.
Sir, myself, Dr. Himakshi Sharma, and on behalf of DDH training team and director Dr. G. Narahari Sastry, we thank you once again for accepting our invitation and delivering a talk in DDH Hackathon training program within a very short period of time, sir. So, sir, we received a lot of questions for your talk, but due to time constraint, we would like to take few questions. Okay, sir, may I start question answer round?
Yes, please.
Thank you, sir. So first question we have, can we get the frag maps and infinity patterns for more than a pair of proteins at a single time?
Oh, that's a good question. In an ideal world where you have infinite computational resources, you potentially can. But for all practical reasons, I would say no. It would make sense to consider building frag maps for each of the protein separately. And I can see where this question is coming from. You want to understand how the affinity patterns are in the context of a protein with its neighbor, right? But again, unfortunately, given the amount of computational infrastructure that you would need for efficiently driving that sampling, it would still make sense to run those simulations separately for each protein partner or each protein chain, if you're considering a multimeric chain, and then evaluate those affinity patterns separately.
Thank you, sir. So next question is, which software is best for protein-protein docking calculation?
There are many options available. I've personally tried out the FT mapper strategy, and which I believe is now available through the Schrodinger suite. So they do a decent job with respect to getting an initial expectation of how potentially two proteins would dock, right? But then again, given the fact that each of those calculations are driven by starting with a crystallographic conformation, you want to again relax a given protein-protein dock pose with at least a small amount of molecular dynamic simulation to see if there is certain change in that interface from what has been predicted using the docking approach. So yeah.
Thank you, sir. So next question we have, are these fragment molecules follows the Lipinski rule and Weber's rule?
They do. That's a great question, actually. So the fragment molecules that constitute the silk simulation have been chosen to be fragment-like. In fact, the current silks mixture, they don't have any intramolecular degrees of freedom. You have benzene, you have propane, you have methanol, formamide, imidazole, acetaldehyde, acetate ion, and methylammonium ion, each representing the different functional groups. And as I rattled off the list, you know that each of them are very small functional groups. So yes, they do adhere the different rules, if you want to call it that, or guidelines. And primarily, again, because you want to get the affinity patterns for these individual fragments and then stitch them all together towards building a more realistic drug-like molecule, which again, you potentially want to adhere to the Lipinski rule or any other constraint that you may have. But the fragments themselves are built to be small for this reason. Yes.
Thank you, sir. So maybe this is the last question which I am taking.
Okay.
The question is, what is the effect of solvation in macromolecule during MD simulation? Can we see any difference if we don't solvate the protein during MD?
Yeah, so you would want to consider some form of solvation. It could be explicit, it could be implicit, but I would still vote for an explicit solvation, primarily to see how the protein responds. Just forget silks as a fragment-based approach. Forget any ligand approach where you're considering desolvation. But primarily, proteins in biology are surrounded by water. So when you strip out the water and just consider a vacuum styled approach, you're already entering a huge amount of artificiality into that calculation. So by that context, yes, any macromolecule, you would want to represent as much as you can the native environment in which that macromolecule thrives. And more often than not, if you're talking about biological macromolecules, yes, it would make sense that you find some scheme in which you can represent the solvation. Explicit water is the best, but if you have computational limitations, maybe consider implicit solvents. But then again, be careful of the kind of conformations that come out in response to implicit solvent versus explicit solvent. Yeah.
Thank you, sir. So thank you very much for your nice explanation for each question. And you covered a very important aspects of computer-aided drug discovery. So I hope your talk will motivate and encourage the students to participate in drug discovery hackathon. Sir, we would like to wind up the session here. Thank you. Thank you so much for being with us, sir.
Thank you. And thank you again for everyone for giving me this opportunity. And thank you for everyone for joining today's call. Thank you.
Thank you, sir.